your talk, and then hopefully that will allow Dr. Luzzatto, Professor Luzzatto, to present after. Professor
Happi, should I bring in Professor Happi?
Yes.
Professor Happi, I have your slides and I'm happy to move them for you.
Oh, okay, great. Well, Christian Happi is my name, and then it's a pleasure to be here and then to discuss this very important topic. And I want to thank Julie for inviting me. And then basically mine is very short because I think Frank has done a great job in terms of talking about the situation in the world and in Africa. So I basically just want to focus on one simple thing that we do, and then which is the genomic of SARS-CoV-2. So next slide, please. Well, as you know, coronavirus is a very large family of single-stranded RNA. So they can infect both human and animals, and then and usually you find them in human. You find about 6 different strains of coronaviruses, and then the most dominant are the ones that the SARS. What we call the severe acute respiratory syndrome coronavirus, and then the MERS that we find mostly in the Middle East, but then we also find them in some areas in Africa. So the new coronaviruses emerge, we say periodically in humans because, and this is basically due to several reasons. One is actually they're highly prevalent, and then the second thing is that they are largely genetically diverse. And because of that genetic diversity, then we also have a very strong level of frequent recombination. And because of that recombination, then we tend to have pretty much new viruses emerging, just like the COVID-19 that we have here. Next slide. So, and for the SARS-CoV-2, which is actually the virus which is responsible for COVID-19, it was first reported in December in China, in a small place in the Hubei province of China, in a town of Wuhan. And then the pathogen, as you know very well, was identified just based on basic surveillance of the disease, of pneumonia and etiology. And because of that, then after probably doing some additional, research and then eventually using, next generation sequencing, they actually realized that they actually discovered a new pathogen, and then we went on with that. By February 2020, WHO announced the new name, which is the SARS-CoV-2 that we see. And then the next year, a few months after then, it was actually WHO called an international disease emergency. Next. So, on March 11, this is a very important date when the WHO declared COVID-19 as a pandemic. So at that point, we had more than 18,000 cases of coronavirus in about 110 countries. But I think these numbers, as Frank mentioned earlier on, has definitely, we've actually over tenfold increase, and this is just within a month. So it shows clearly that this virus is very different from the other coronaviruses, and then also is spreading very fast. Next. So this basically just show you globally the epidemiology. Then you see the case fatality. You see the number of cases and then the case fatality. If you look at these two maps, actually they match very well. And this definitely shows that as the number of cases increases, then we're definitely going to be expecting more death. And I think Frank has done a very great job by actually telling us exactly, where we are, and then how, we tend to think that in Africa, the epidemic is different. Absolutely. I agree with him that the epidemic is not different. And I'm very, very actually worried that, the case in Africa, if care is not taken, we may end up having a very worse scenario. So, and this very point to the fact that you have a very strong correlation between the number of cases that you identify and then also the number of cases of death. Next. But then the question is, this gives you the case, I'll pass through this very quickly. So this really gives a highlight of what happens. This is older than Frank presented a slide that is much more recent. I think this is a more older slide. But the point here is actually to show that, you know, why we in Africa, we thought that we could be in a situation whereby, you know, the virus was spreading slowly, but at least it's clear that, you know, the virus is actually virtually everywhere on the continent. And then we also have cases, now we have cases of community transmission. And then this becomes very dangerous when we start having community transmission, because remember that I mentioned the fact that when you have these viruses transmitting on regular basis, that's where we have, you know, a higher combination rate within the population. And then that really leads to a situation whereby viruses become very difficult, you know, to track. The diagnosis become very difficult, you know, I mean, to perform. Performance in the sense that, you know, this diagnosis, the molecular diagnosis that we develop are often based on specific target. And then those targets, if we do have mutations, then we think that, you know, it might be very difficult to actually use even the tool that we have now to identify these viruses. And then also it becomes a potential danger for the other countermeasures. This includes, you know, therapeutics and then also potential vaccines. Next. So, and what I wanted to just dwell on a little bit is the fact that, you know, genomics over the years has become a very important tool in terms of tracking, you know, the spread of infectious diseases. We basically started the first application of genomic epidemiology during the Ebola outbreak in Africa, in West Africa in 2014. And that was the first time where we basically, you know, in the first 3 weeks of that outbreak, we sequenced 99 genomes and they made them available to the public, I mean, to the international community to use. But then over time, we were able to use, you know, the same type of information using the genomic data to actually show, you know, transmission and then spread of a transmission and spread of the outbreak over time. And this really will learn a whole lot through the process. And through that process, we're able to see, actually to demonstrate different clusters and then showing transmission, or community transmission. But apart from showing community transmission, we're able to show how the virus evolved over time. In the case of Sierra Leone, for instance, we're able to show that, you know, we have 4 different strains of Ebola that got into Sierra Leone at different time points. But then the map that you see here, for instance, shows across the globe how, you know, the virus moved from one point to the other. But apart from showing how the virus is moving from one point to the other, then you can see the different clusters with different colors. Different clusters and different colors actually here will show you how you can have transmission, you know, clusters, I mean, showing the transmission occurring within different communities, but also showing the relationship between what you see in some place in different parts of the world, and then seeing how the viruses move from one area to the other. But exactly what actually is responsible for the movement of viruses, the viruses doesn't just fly. I think we humans actually use, I mean, are the vehicle for movement of viruses across the globe. But then the point is that once the virus move and then get to different places, then there's also the virus is subjected to different type of selective pressure. And that absolutely affects the genetic diversity of the virus or the evolution of the virus over time. So, and this process really makes it very complicated. So this slide here gives you an idea during the phylogenetic and phylogeography of the SARS-CoV-2, you know, I mean, between December and then March 2017. But then, as we mentioned, we had the first case of COVID-2 in Africa, I mean, by mid-February. And then the first case in the country where I live, Nigeria, next slide, occurred in the first weeks of March. But what is very interesting, I mean, later in the month of February, that was around February 27th. And what we did was really to go on and characterize, you know, this virus very quickly, because the idea of using this stemmed from the fact that we work in very close collaboration with the National Center for Disease Control, and we've been able to use over the years, you know, genomic technologies as a way not only to characterize pathogen, but also to uncover, you know, unknown circulating viruses. And this, for instance, in 2019, we uncovered a new strain of yellow fever circulating in Nigeria, but then at that point, the diagnostics for yellow fever wasn't working. And we eventually used metagenomics as a way to identify a new strain of yellow fever, inform the government, and then they declared an outbreak and they were able to contain that. And dating this strain, we realized that this strain was very different from the other ones that have been circulating in Nigeria for the past 100 years. So, and then this shows you again how this virus is, what I'm talking about here on this particular slide shows how the virus is spreading so fast. Within a month, we went from 1 case in Lagos to about 254 cases, and then the distribution in Nigeria is so widespread. It's virtually in all corners, and in a large country like Nigeria, more than 200 million people. So we really sitting on a taking, on a time bomb, and we're very worried by the fact that if this spread out of Nigeria, even though the country is locked down, but we just imagine this number of people within this country. If this continues to spread, then we may end up having a very dire situation in our hands. So next slide, please. So, and basically what we did at that time was actually to work with the Nigeria Center for Disease Control and then the Lagos State Ministry of Health, who actually had the first patient. And we were able to use one of the 2 Illumina mice that we had in our lab, to really have a full genome of the SARS-CoV-2. And this was done, in a very unprecedented manner because we did this in 48 hours. So within 48 hours, we were able to have a whole genome of a virus elucidated, and we're able to trace it back to the fact that yes, although the index case was from Italy, but then the virus really clustered not with most viruses in Italy, but they cluster mostly with viruses in Switzerland and then in Finland. Further investigation actually, proved us right, because it was clear that this Italian citizen actually spent so much time in Switzerland before coming to Nigeria, and then that was where he contacted, the infection. So clearly demonstrating how we could actually use, use this information as a way to show clearly the origin and the source of the virus. But then what is also good about this is that we generated here the first genome for Africa, but then apart from generating the first genome in Africa, we also made it available in GISED and then contributing the first genome of Africa into GISED, and then bringing that also contributing Africa's science to the global community. Next. So
I thank you. 3 more minutes, Chris.
Sure.
And
this is my second to the last slide. So this basically just shows you what I've described earlier on. You can see this particular cluster where you have the Nigerian sample. Clustering very well with those from Switzerland and then Finland and other countries. So here you also see the coverage, across the coronavirus as a genome. So pretty much, I just feel that, this was for us a very proud, a moment of proud, having done this within such a critical time and in such a short time to contribute Africa genome in the community of, in the community of SARS-CoV-2 genome. So, and this basically just shows us how we could actually leverage genomic technology as a way to not only to do science, but also to contribute, to public health intervention. So what we're doing right now, we're doing a massive genome sequence, about hundreds of samples, not only from Nigeria, but also across the continent through the Africa CDC. We have other countries who don't have the sequencing capacity, sending this down to transmission across different countries with the hope that we're going to elucidate actually COVID-2 epidemiology or genomic epidemiology on the African continent. I think on this point, I do think that this was my very last slide. And then at this point, I really want to thank you for the opportunity, and then also thank Julie for organizing this meeting. Over.
Thank you very much, Chris. So that was Professor Christian Happi, and it was a terrific talk about the genomics of COVID-19 virus and the work that they've done in Nigeria. Whilst we're waiting for Lucia to call in, so Lucia